Okay, thank you so much. I am kicking off this string of exosome conversations, and I promise that by minute 6, I will bring back up exosomes. We are actually a personalized regenerative medicine company, or Acorn Biolabs, and ultimately, as many people have experienced this, we are in the beginning of an anti-aging revolution. The vernacular and the words that patients are using as they come into clinics are now using things like exosomes, PRP, PRF, knowing the difference between those things, talking about stem cell-derived applications, and the education has been increasing and the demand for these regenerative medicine treatments, especially around words like anti-aging and longevity, has exploded. We focus on the part of this that is powered by your own cells. I've experienced this twice now in my career, once formerly as a professional athlete many years ago. I saw members of my team literally striving to have access to PRP, PRF, all of the latest and greatest applications to really return to sport faster and elongate their careers. On the other side, we have seen red carpet celebrities make things like the vampire facelift popular, including where we are all the way up in Canada. We hear about it too. The issue is that generations, unfortunately, are going to miss out on the potency and the potential of these applications. And that's because every day that goes by, unfortunately, we are getting older. Our cells are becoming a poorer and poorer resource. And if we're using our own cells to create these, then unfortunately, the therapeutic benefit and that delivery is going to diminish over time. I learned this firsthand, actually working on connective tissues at Mount Sinai Hospital and developing applications for us to grow them on demand for patients. The first project we were working on was cartilage. My role going into that group was actually to take some very successful animal studies that had been conducted on cartilage regeneration and translate that into human models. All of the animal studies that had been conducted were on juvenile animals, so basically teenagers. And now I was going into the OR and taking a biopsy of patient cells at the time of arthroplastic surgeries, so fake knees and fake hips. As you can imagine, the demographics were vastly different. And after a lot of trial and error, what we really understood was that age was the greatest influencing factor on our ability to create functional tissues to deliver back to those patients. And that is, unfortunately, pretty limiting when you're looking at trying to deliver things to patients at a time of need. Because when we go in and we seek those things out, we're usually later in life, chronic wear and tear has kicked in, and unfortunately, our cells are performing at their absolute worst. So we had a bit of a roundtable thinking about what we could do, and a few big things came up on focusing on extracting cells non-invasively, painlessly, and being able to get a high-quality cell type that we could actually get in advance of that aging process and bank that patient's cells at a younger time point. And I'm excited to share with you today Acorn's live cell preservation service. This is a simple way in clinic to be able to offer an opportunity for patients to take samples from their hair follicles, plucked from the back of their scalp, simply and easily, and access the MSCs that reside in that hair follicle and cryogenically preserve them for the future, locking in time and age. You did hear that right. We focus on the hair follicle, and we love the hair follicle for many reasons, but 4 big ones. It's a rich source of cells. We have keratinocytes, fibroblasts, and mesenchymal stem cells within these hair follicles. They're highly expandable. We can make more of them on demand, and they're reprogrammable. So in some of these future strategies, we see them really being able to use in reprogramming technologies to tackle some very big diseases. They're collected non-invasively and painlessly, so from a patient perspective, they love it. In fact, it's a lot easier than trying to get blood out of them. And from a physician perspective, these cells are a patient's own cells. So we're not worried about immune rejection, putting a patient on any drugs for immunosuppression, or with skin, thinking about things like graft versus host. Acorn is not only your cell preservation resource, but we're actually also actively developing the therapies that are going to be taking those banked cells and leveraging them for patient benefit. We think of this space really across 3 timelines. Long term, it is going to take very long regulatory considerations and trials, but are tackling some very big things like Parkinson's and macular degeneration. In the medium term, we're thinking about using the live cells themselves and reintroducing them to the body to enhance the youth and the performance of those cells. And then in the short term, it's leveraging those cells in the lab to create molecules, exosomes, matrix molecules, as well as growth factors that the patient can then retain from their younger selves and continue to deliver back to themselves over time. The first application that I'm talking about in that very short term, and we're going to be going into first human applications this year, is an actual derivative of that patient's stem cells that are stored. They're taken out of the fridge, they're cultured in our laboratory, and they're allowed to grow and produce exosomes, growth factors, and matrix molecules, releasing those into the media. We can actually harvest those every 2 days and distill that down into a conditioned media that can then be delivered back to the patient, lyophilized first, and create a series of vials that can be delivered to that patient over time. So from that one instance of collection, you're creating many, many applications that you can deliver to that patient over their lifetime. We've done 2 preclinical studies on this. The first one compared it head-to-head from PRP that was generated from the same patients. I was excited to say we were able to create a number of the macromolecules of interest and growth factors, including fibronectin at 34 times higher volumes, all of the growth factors between 2 and 26 times higher volumes, and on and on. So we really like this as an alternative to PRP, right? And some of the other regenerative medicine strategies. By the way, these volumes were both free proteins and matrix molecules, as well as those that were contained in the exosomes produced themselves. The next thing we did is actually run a performance study. So we took a patient's cells and we cultured them in the laboratory and allowed them to cover the sheet and reach confluency. We then created an artificial wound by taking a scalpel and running that along the skin surface. And in one side, we ended up growing the cells in a standard growth media. On the other side, we added 10% of our serum to that growth media. We were able to enhance the closure of that artificial defect after 48 hours by 3.5 times. So we really are excited about the performance of this media being applied to cells that are live and growing. Like I said, we're not just an exosome company. We're not just even an aesthetics company. We are looking at some of the longer-term strategies. And so a lot of this work was done in collaboration with University of Toronto, National Research Council, University of Calgary, and University of Ottawa. But to dive in, this is actually my 13-year-old son's cells, all under REB. So no questions there afterwards. But we were able to create from their hair follicle their own fat, bone, and cartilage on demand. Now, he's not considering a BBL just yet, but as a little basketball player, he was very excited to see his own bone and cartilage growing in a dish. On top of that, we can create neurons on demand, including dopaminergic releasing neurons to target things like Parkinson's. And even beyond that, we can reprogram these cells and push them down all 3 different germ layers. We picked pancreas as an example because my brother happens to be a juvenile diabetic, and we were able to create pancreas cells on demand from the patient's hair follicle. So pretty exciting work, again, long-term strategies. Acorn is available in Canada. We launched in April, and we're very excited to announce that we've built out our facility in the Los Angeles area and have started already servicing Southern California. We're opening up our first sites throughout the spring across the US, and very excited to engage with physicians that are interested in learning more about our technology or interested in participating in some of the trials and the applications that we have going on in aesthetics and wound care. Thank you so much for your time. I really appreciate the invite here, and that's, I did mention exosomes, so thank you.